Okay, so now that we've talked about a little bit of a review of the cell, the organelles, the plasma membrane, and the general ideas with the nucleus and what could happen, the reason you want to review those things is because you really want to know why they're there. If you understand why they're there and you have a malfunction to any of those organelles, you can see signs and symptoms that are associated with it. So the next thing we're going to do is we're going to talk about what happens when you alter the cells, when you change them. And there are a handful of things that you can do to a cell. So a cell can be changed by injury. It can be changed by adaptation, changed by aging, neoplasias, or you can think of cancers, and then death. So there's a limited number of causes of disease or things that can happen, and we're going to go through the main causes so that when we go into specific tissues later or specific systems, you can reference back and say, well, I wonder how this happened, what type of organelles are in there, what could have happened possibly to cause these alterations or injury. And I look at it kind of like your engine. I mean, I used to work on engines when I was in high school, and there's a limited number of things that could happen to an engine that keeps it from running. I mean, once it's running, you know that there's gas going, there's oxygen going in there, there's a spark that's causing the cylinders to push, and there's exhaust coming out, and it stays running. Fine-tuning it from that point. But if you deprive it of gas, it stops running. If you deprive it of oxygen, it stops running. It's amazing because where I'm from in Illinois, we have flooding right now. And there are people that try to drive their car through the water, and as soon as the car can't get oxygen, it stops running. It stalls. Just like us. If we don't have oxygen, we stop. You need a spark. You need something that triggers the whole reaction. You need a cylinder that causes the movement, and then you need exhaust. It's like in the old movies where they shove a potato in somebody's tailpipe and the car wouldn't start because the exhaust wouldn't leave. It's the same idea. If you break any of those things, the engine doesn't run. If you change or alter the cell in a certain number of ways, it'll keep it from running. And we're going to talk about some of those injuries first. So the injuries are going to fall into these four categories. Either there's a deficit injury, so hypoxia is the #1 type of deficit injury. Ischemia, malnutrition, dehydration, all are depriving the cell of something. Toxins get in, and toxins can be from something made inside the cell, which is endogenous, or something that's outside of the cell, like a drug that you introduced or something a bacteria brings in. Infection means that something infected it, either a bacteria got in around the tissue and started attacking it, or a virus actually got into the cell and caused it to lyse or break apart. And then physical injuries, and I'll just talk about a couple of those. So they can be temperature-based, they can be mechanical, like smashing, squeezing, and we'll talk about how those work. So let's go into the deficit injuries first. So when you look at deficit injuries, you want to look at things like carbs, fats, and proteins. So forms of malnutrition. If you don't get enough carbs, you get hypoglycemia. You don't get enough sugar, which means that you're not providing enough of this raw material to make ATP. Think of things like neurons. They're going to shut down. The heart's going to shut down. You have to have enough sugar. The cell needs it. It's like the gas for the cell. Fats are the same way. Fats are important for building structures like membranes around the cell. We already talked about that in the review section. If you don't get enough things like cholesterol, you can't make the proper membranes, and the cell will fail. It doesn't adapt to temperatures as well. Proteins, just protein is a basic raw material. It builds a cytoskeleton of the cell. If you don't have enough protein, your body will actually break down protein from other areas to provide amino acids. And then things like vitamins and minerals. Vitamins are cofactors. They work with other enzymes to help the enzyme work. So if you're missing the cofactor, the vitamin, the enzyme can't work properly. It's almost like having the proper key to start the enzyme. If you're missing the vitamin, it doesn't work. And a lot of vitamins, like vitamin B12, is really important for helping DNA replication. We'll talk about some of the different vitamins, and actually I'm going to give you a small assignment that you're going to look them up. But just as one example, vitamin A. Vitamin A is important for maintaining retinal receptors, so vision. It helps develop red blood cells, white blood cells. It helps maintain bone density, so it has a lot of functions. You need vitamin A. If you have a deficiency, if you know you need it to help with things like retinal development, it's going to cause blindness issues. So night blindness, just permanent blindness. It can cause dry cornea because it's not nurturing the eye. If you don't have enough and you can't properly develop or build white blood cells, it's going to decrease immunity. It's going to make you more susceptible to mutations and things like cancer. And then it also causes things like birth defects. And I don't want to stick with just the deficiency. You can actually have an excess of anything in the body. Too much of anything, whether it's a good thing or a bad thing, is too much. Like carbs, you can get too much carbs. You know, if we can't regulate the carbs in your body, you get something called diabetes, or actually I should say diabetes causes that irregulation. Lipids, you can have too many lipids, like fats. Fats are really interesting because if you have too little, you can't build cell membranes. If you have too much, then you start having all these fats sticking to like the walls of the arteries, clinging and causing atherosclerosis. In fact, I'll show you that on the next slide. I'll talk about it a little bit more. So you need fat, but too much can actually cause problems. Vitamin A is the same way when you talk about vitamins. The fat-soluble vitamins can cause toxicity. The water-soluble ones are easy to get rid of. You eat too much vitamin B, which is water-soluble, you just pee it out. You know that because when you take a B vitamin supplement and you go to the bathroom, your urine's yellow after that point. But the A, D, E, and K are the fat solubles, and any of those can actually cause toxicity. So like vitamin A, getting too much actually causes your cells to swell. It puts your cells in this expanded hypoosmotic situation where they start swelling up. Fat-soluble vitamins have a tendency to accumulate in the liver, but when the liver's full, they float through the blood and they find other fatty tissues, like the brain. And since they love fats, they cross the blood-brain barrier and they can actually cause toxicity in the brain. And then another bad thing is when we talked about vitamin A being important for maintaining bone density, if you have too much vitamin A, it overstimulates osteoclasts. And if you remember your anatomy, osteoclasts do what? Break down bone or build up bone? They break it down. So too much vitamin A will actually cause your bone density to decrease because it's breaking it down via osteoclasts. And then too much vitamin A actually blocks vitamin D. And what does vitamin D do? It helps you pull what mineral in from your GI tract? Calcium. So if you have too much vitamin A, it actually prevents you from absorbing calcium, which means you're going to break down more bone to put that calcium in the blood. So it's really an interesting pathway. You can have too little or too much of certain things. This was just an example of the hypolipidemia versus hyperlipidemia. So when you're not eating fats, what happens is almost like you're in a starvation state. Your fat cells will start releasing fatty acids that go to the liver. And the liver will start changing them, manipulating them, and processing them. And they build something called ketone acids or ketone bodies. Those ketone bodies can be cleared by the kidney, but they're also acidic, so they cause a toxic environment. Hypolipidemia can also cause accumulation in the liver because when it's processing too many fats, it keeps too many of those fats in the liver and causes a fatty liver. At the same time, you can have hyperlipidemia, which means you make way too many or you eat too many lipids. And those lipids go through your circulation, they bind to the walls of your arteries, and they stick there. They're fat-loving. They can seep right into the wall of the artery, and then your immune system has to come along, like these macrophages, come along and they try and eat it and clean it up, and they die in there. So they just accumulate, and they get thicker and thicker until your arteries become blocked, and that's when things like heart attacks happen. So cells can have too much or too little. Overall, the whole body can have too much or too little of anything. So the first homework question in this set, or homework question number 8, is that write down vitamin deficiency, each of these vitamin deficiencies, and then label what vitamin is the cause and one prominent symptom. So one symptom that helps identify, like, let's see, not scurvy's on there. So is rickets. I'll just give one away. So with scurvy, when you see symptoms of scurvy, the vitamin, I'm going to have you look it up, the symptoms are actually because this vitamin that's really important for you helps build collagen. It helps hold your skin together, gives strength to the skin, helps hold like the gums and teeth together, helps build blood vessel walls. So a prominent symptom will be blood vessel issues or breakages of blood vessels. And you can see it a lot along the gum lines. You can see it along the skin. So they call it petechiae, little breakage of capillaries. So a visible sign. And you probably heard of scurvy because of the old pirate stories and stuff. But go ahead and pause, write this down, and then do your web search for it. All right. The number one deficit is oxygen. And make sure you know the difference between the terms where hypoxemia and hypoxia are not the same thing. Ischemia causes decreased blood flow, which means decreased nutrients and decreased oxygen. Hypoxemia just exclusively means decreased oxygen. The emia in both these words says it's where? In the blood. Hypoxia is referring to the tissue. So hypoxemia is decreased oxygen in the blood, and it can be caused by things like not enough oxygen in the air so you can't grab a hold of it. It could be not enough red blood cells so you can't hold on to oxygen. Hypoxia is talking about the tissue. So the rest of the body may have plenty of oxygen, but that tissue is being deprived. So you want to think of things like a blockage. Maybe you have a clot that went downstream and blocked off blood flow. if the blood flow is not getting to that tissue, the tissue will become hypoxic, which means low oxygen, and then anoxic, which means completely without oxygen. So here are some examples. And this last one here, ischemia, decreased blood flow, can be caused by things like atherosclerosis, or that narrowing we talked about on the last slide. It can also be caused by a blood clot that sticks in the blood vessel and causes the damage. We're going to talk about this for just a second. In your book, if you look up myocardial infarction and the steps of it, these next slides are actually going right along with it. So here's an example of what would happen from ischemia progressing on to a heart attack or myocardial infarction. So the first thing that's going to happen is that you've got years of this accumulation in your arteries, we'll say. So here your coronary arteries have this atherosclerosis. Then you have a blood clot that's thrown. The blood clot comes along, sticks in here, and over the first minute, you're deprived of oxygen, but this heart muscle actually has something called myoglobin in it that holds oxygen. So it can keep working for about that first minute. But now the myoglobin's gone, you're not getting fresh hemoglobin, so after that first minute, there's less aerobic metabolism. Remember, aerobic metabolism refers to the mitochondria making energy. 1 sugar turned into 36 ATP, general rule of thumb. So since you have less aerobic metabolism, your ATP decreases. Makes it harder for the muscles to contract, because remember, the muscles need ATP to contract and for them to relax. So you're not going to get the same contraction. After about 3 to 5 minutes, you're completely anaerobic. There's no oxygen left in that heart muscle. You're depending on what process to keep the heart contracting at all. Glycolysis. But remember, glycolysis only takes 1 sugar and turns it into 2 ATP. Very inefficient. And it also makes a lot of that lactic acid. When you're accumulating the acids, you're going to do a couple things. You're going to start denaturing proteins like pump proteins, calcium pump proteins. You're also going to start turning off the ATP for these pumps. When the ATP is turned off, the pump doesn't work either. So now suddenly your pumps don't work. The pump, remember, was important for pumping sodium out of the cell, 3 sodiums, and 2 potassiums in. So now the sodium's leaking in at a fast rate, potassium's leaking out at a faster rate. What's going to chase the sodium? Water does. So water chases the sodium in. What's going to happen to this cell? It's going to start swelling. It starts getting bigger and bigger. And just like what I referred to earlier, the cell's like a clock. Every gear has to be in perfect place. If you swell the cell, the gears don't line up anymore. In fact, what happens during cellular swelling is that the ribosomes start popping off the endoplasmic reticulum, which means you can't make proteins anymore. The mitochondria, which they don't have in this picture, starts swelling. And the mitochondria is going to start tearing and damaging that cristae. So those little weaves in the mitochondria that are important for energy production. So even if you get oxygen back, the mitochondria is not going to work the same because the assembly line is actually disassembled. So you've got lots of problems going on in here. You can think of the Golgi apparatus is swelling. All the structures are swelling. Now you're getting all these little water vacuoles in here. It's called vacuolation. So now water is building up in little pockets, causing even more pressure on the inside of the cell. So you can see this is a bad situation. And this is in the first 3 to 5 minutes, you're starting to shut off these pumps. You get swelling. And then here's the problem. This part we don't completely understand is that if you provide oxygen back to the cell, so you give this person high concentration oxygen, you clear the blockage, and now oxygen is getting back to the tissues. There's something called reperfusion injury. It's kind of a mystery, but what we think is happening is that when the mitochondria's assembly line is actually broken, it doesn't want to come back together. So if you start giving it oxygen and sugar, it tries to make ATP, but it does it very poorly, and it actually makes more of a nasty byproduct called reactive oxygen species. So I've got this little example. It's kind of like if you put the wrong type of fuel in your truck and then you try to put the right stuff in later, you're going to get all this nasty byproduct, this toxic waste shooting out of it. And that's what the cell does. Instead of making good energy, it's going to sputter along, making this really toxic form of energy. So let's talk about those free radicals. I know you've heard of them before because when you watch any kind of television that has a commercial for like makeup products, they talk about the antioxidants in makeup products that neutralize free radicals and slow the process of aging. Free radicals are destructive little atoms or molecules. And if you've had physiology with me or you've had anatomy with me, I always talk about this. But free radicals, I'm just going to use one example. I would say that if hydrogen is the one I usually use. So hydrogen has 1 electron, and it's not in this picture, but hydrogen would be this little atom here with just one electron, one of these yellow things floating around it. It's the only thing it has out there. If you take away its electron, now it's actually very toxic. It's angry because you stole the only thing it had in its world. So it goes along trying to harm other ones. When it's a hydrogen that does that, we call it acid. So the hydrogen ion that has a little negative charge over, or a little positive charge over it, is actually a type of a free radical, basically because it's very toxic and very dangerous, and we call it acids. Other cells do the same thing. So if you take other atoms do the same thing. If you steal an electron from it, it makes it very angry. It's ticked off. It's going to go to its neighbor, and it's going to steal an electron back. But now that neighbor is ticked off, so it'll steal one from its neighbor, and then from its neighbor, and its neighbor, causing a chain reaction. And once they steal this electron back, they don't get any happier. They're actually changed, they're deformed in some way so they don't work properly anymore. So what happens is if this free radical is made by the mitochondria and attacks the lipid membrane, it's called lipid peroxidation. And then it's going to deform the whole membrane. What if it punched a hole in the membrane? What would happen to that cell? Right, it would lyse, it would pop. Right? The membrane's supposed to be intact. What if it changed the proteins, like denatured the proteins? This angry molecule changed the proteins, and now the pumps don't work. Now it doesn't have cell recognition like it had before. All of the processes, like enzymes that proteins are important for, none of those work anymore. So the cell's doomed to fail because of these free radicals. It can get into the nucleus, and it can actually change the DNA. It can chop apart the DNA and destroy it. That's one of the ways that mutations happen that could lead to cancers or cell death. And then, of course, the mitochondria, any of the membrane-bound organelles, mitochondria, endoplasmic reticulum, Golgi apparatus, they're susceptible to attack from free radicals. What's interesting is that the mitochondria is usually making these free radicals in the reperfusion injury, and then it comes back and attacks it, making the situation even worse. So free radicals are important. And like I said in the beginning of this slide, cosmetic commercials sell antioxidant products to slow free radical progression. Your body actually makes antioxidants, and an antioxidant is just an atom or a molecule that has extra electrons. So it finds a free radical and says, hey man, you can have one of mine, and it shares with it. So it stops that angry chain reaction you see here. And the antioxidant doesn't become dangerous because it had plenty of electrons, no loss to it. So that's how free radicals work. Free radicals cause lots of problems. And this is actually a picture from your textbook where you can see here's a normal cell. Another site of free radical production is called a peroxisome. It makes something called peroxide. So here you have hydrogen peroxide, H2O2, a little different than water. Right? So this hydrogen peroxide is really useful because it denatures proteins. It breaks things apart. If you put hydrogen peroxide on your hair, it changes the color of the hair, it changes the structure of the hair. Back in the 80s, I remember when people would actually use peroxide on their hair, it made their hair real dry and frizzy and gave them a lot of split ends because it's really dangerous and toxic. When you put hydrogen peroxide on a cut, it kills the bacteria because it does the same thing to a bacterial cell that it does to your cells. It goes along, destroys the membrane, it destroys the DNA, it attacks any of the structures on the inside. Bacteria doesn't have organelles, but it attacks the RNA and the other structures like enzymes that are important for reproduction. So your body luckily makes these things called antioxidants, superoxide dismutase. You're not going to have to remember these names, catalase. But it makes these things to neutralize free radicals. Unfortunately, when free radical production goes out of control, your body can't make enough superoxide dismutase or catalase, the antioxidants, to slow it down. And that's how reactive oxygen species work. And you'll want to know how those processes work too. Just in general. All right. Next injury could be due to toxins. And I know there's a list of toxins, and don't get freaked out or spooked. In general, this is how a toxin works. A toxin gets into the cell, and remember the cell's like a fine timepiece, like a watch. It gets into the cell and it acts like a piece of sand. It gets in between the gears and blocks the gears up. So in situations like gases like carbon monoxide or cyanide or hydrogen sulfide, carbon monoxide gets in and it binds to hemoglobin and red blood cells, and it bends the gears so that oxygen can't get in there. It bends it just out of shape so that nothing else can get in. It stays bound to it. With cyanide, this stuff's so potent it can kill you in 30 seconds. And a similar chemical called hydrogen sulfide is made by you. You actually make this in your body. Bacteria makes it. And this hydrogen sulfide can actually get in, and what it does is it blocks up enzymes. So the enzymes don't work. And it stops them. What's really nasty about hydrogen sulfide is that it stinks really bad because it has sulfur in it, but it only stinks for a few seconds to a few minutes, and then it numbs your nose so you can't smell it anymore. So you're breathing this toxic substance in that can be killing you the whole time. Heavy metals like lead, mercury, same idea. They get in, they block enzyme reactions. So lead and mercury typically go to the mitochondria and they stop it from making energy, which can kill the cell really quick. Ethanol, this is, something a lot of people enjoy on weekends, can actually become toxic. And I'll show you a slide, the next slide, that covers it a little bit better. And we'll talk about things like social drugs or street drugs like cocaine and how they affect you, but we'll talk about that more when we get to neuroscience. Right, and then this last one, carbon tetrachloride. This is really interesting because this is a solvent for cleaning grease off of car parts. And if it gets on your skin, it seeps in, goes to your liver, and will actually block your liver's ability to process fats. So fats get into the liver, they're stuck there, and it causes this really fatty liver. And within minutes, it can cause lipid accumulation in the liver, and your liver expands. So it starts swelling up. And within hours, it can actually kill you. And if they took your liver out, you've got this real slimy liver because it's so full of fats, they can't get processed properly. And that's what carbon tetrachloride does. It gets in here and it blocks up the gear so your liver can't process fats, and they just all start sticking in there. So let's talk about alcohol just a little bit. This is kind of interesting, and you don't have to memorize any pathways. When you look at alcohol, let's say you take a shot of tequila, and when you drink it, that shot of tequila goes to your liver. And it's processed through a couple steps of enzyme reactions, and it's turned into actually something called acetate, which is basically the same component that makes up vinegar, acetic acid. So, of course, acetic acid's acidic. It's not really good for you. It would be like you drinking vinegar. But your body's making this stuff, and it can process it just fine. However, if you take in too much alcohol, all these little enzymes that process the alcohol don't work. So your body has to make another pathway. And unfortunately, this pathway, look at all these branches. These branches are responsible for making lots of free radicals, reactive oxygen species, which can damage the liver cells. They can get through the body and damage other cells in your body, like your brain, your kidney. They can get into your GI tract and cause problems. These chemicals, these free radicals, can cause lots of damage. Having one strong night of drinking isn't going to cause permanent liver damage typically, but it takes a little time to recover. Consistently drinking a lot of alcohol or consistent drinking excessive alcohol can cause a lot of damage to the liver. Luckily, the liver is pretty resilient and can come back when you stop drinking, but if you cause permanent damage like sclerosis or scarring of the tissue, then it doesn't come back. So just something to keep in mind. All right, infectious agents, bacteria and viruses are the primary infective agents. So bacteria can get in, and bacteria can go around, and it likes to make a home in your body. For instance, the malaria parasite. Malaria gets in, it goes through a mosquito bite into your body, and then it starts developing in your liver. Then it starts releasing more of its little parasites into your blood that go to the red blood cells. Once they're in there, they start just eating, having a good time, and then eventually they lyse the red blood cell or break the red blood cell, causing anemia, causing you to die. Another one, viruses. Actually, I'll show you this picture. Viruses get in, and the way viruses work are they can't replicate on their own. They're not a real cell. They get into your cell, take over the machinery, they replicate, and then they make lots and lots of little tiny viruses until they fill the cell up, and then the cell lyses or breaks and dumps all those viruses in the environment. So your cell actually is killed while these are giving birth to baby viruses. So that's another way that infection can attack your cells or injure your cells. And then this last one here is how your immune system can actually attack. And your immune system, if it's attacking your own cells, is called a what kind of disease? Immune attacking self. Autoimmunity. These are autoimmune diseases. Poison ivy we'll talk about in better detail in inflammation and immune system, but the little chemicals that are in the poison ivy will actually bind to proteins in your skin. Well, these look foreign, but now they're bound to you. So your body looks at this stuff and says, hey, you don't belong there. And it starts attacking these little tiny products, but inadvertently attacks the skin proteins at the same time, causing death to the skin tissue, peeling, flaking, inflammatory response like swelling, and that's what happens. This can also be seen when you're looking at something like MS. So you can see all these immune cells. So you've got T cells and B cells, macrophages. These, when they get across the blood-brain barrier and they recognize the myelin sheath in there, they say, hey, you don't belong here. even though it really does belong there. And then the immune cells start attacking the myelin sheath until they've destroyed it. So once they attack it, they bind it with antibodies, and then they basically just beat it to a pulp, and they destroy the tissue. And we'll talk about that more when we get to multiple sclerosis in the neuro section. Right. Oops. Totally forgot about this one. So the last one, the physical injury. And this is kind of an interesting one. I'm only going to talk about a few, like temperature is a key physical injury. When you heat up a cell, it's full of liquidy like proteins, and then they cook and they become denatured. They change their shape, which means they don't work the way they used to. It's just like an egg white. It's a liquidy protein, but when you heat it up, it turns into a solid protein, and it doesn't have the same functions it did before. Over here is a cancer cell that's been frozen. You can see that water expands when it freezes, and then it turns crystallized and it punctures through the cell. So in this situation, you can see the nucleus of the cell membrane. You can see all the water crystals that poke right through the membrane and destroy the membrane. So freezing or heating can cause physical disruption of activity in the cell. And this last one I'm going to talk about a little bit is UV radiation or ionizing radiation. It gets through the cells, goes straight to the DNA, and changes the DNA. Once it changes the DNA, it may make it so that that cell can't replicate anymore. It may shut off important proteins for life. And if it shuts any of those off or causes mutations, it can either die or it can become a cancer cell. Right, here are some of the manifestations of cell injury. Here's an easy one because you can actually see it. Let's say you're at a party on Friday and you see some people getting into a fight and one guy gets punched in the face. Immediately within a few minutes, you'll actually see a lot of redness in the area because they've destroyed the capillaries, and now all those red blood cells are seeping into the tissue. Once they're in the tissue, they can't be re-sucked back into the capillaries. There's too much pressure in there. So they sit there. While they're seeping into the tissues, they look really red. Why do they look really red? Why does the tissue look red? It's because the red blood cells are going out there carrying oxygen. While red blood cells oxygenate, it looks very red, so it gives that red appearance. It gives that swelling and that red color. But the next morning when the guy wakes up, what happened? Why is his tissue blue or purple? It's because of the red blood cells. What happened to the red blood cells? They lost their oxygen. When they lose oxygen, they become deoxygenated and they turn more of a dark, like bluish, purpley, burgundy color. And that's a sign of bruising. But now these cells are where they don't belong. Red blood cells don't belong in tissue. They belong in the capillaries or the blood vessels. So now your immune system comes along and starts cleaning that up. The immune system starts eating those red blood cells and spitting out the debris. So it's going to leave a couple things. One's called hemosiderin, which is part of the hemoglobin molecule. And another part's bilirubin. Well, technically it's biliverdin, which is basically pre-bilirubin, which gives it kind of a yellowy, greenish color. So the first day, you're going to see the redness. The next morning, you're going to see this bluish-purple deoxygenized hemoglobin. And then the next day, you're going to see accumulation of these byproducts, these waste products, like biliverdin, the bilirubin precursor. And then the last days, you're going to see it turn a little bit more yellow and then eventually fade away because the tissue's been cleaned up. But there you can actually see the breakdown of cell injury, or the process of cell injury. We talked about this already a little bit. I just didn't give it a name. It's called hydropic degeneration. And that's when you get accumulations of water inside. So here you have this vacuolation, or these little pockets of water that are causing swelling. Those pockets of water don't go away. Once they're in, they're always there. So once the cell hits the point of no return where there's too much water in it, it's committed to die. It doesn't have to be just water, though. It can accumulate too much sugar, so glycogen, too many proteins, some pigments. We'll talk about these later. It can actually accumulate too many lipids, so maybe too much cholesterol in here, and that would be an example with that fatty liver. Too much lipid starts accumulating. We'll talk about this again with Tay-Sachs disease, where lipids accumulate in neurons of children, and it causes central nervous system degeneration. So it can be lots of things that accumulate, and that's called cellular accumulation. As you can see those under electron microscopes, the little accumulations of pockets. Sometimes you can actually see it in a light microscope too, with water pockets or fat pockets like this. Here's lipid accumulation. You can see the lipid pockets that are all over in this tissue. So this is an example of a fatty liver. Some of the causes here, and this is the most interesting one, is that starvation actually causes a fatty liver. The reason is because when you're starving, the liver's trying to process fats to use for energy. And some of those excess fats start accumulating, and they start building up in the liver. And then the liver starts expanding and swelling. And you know this because when you've seen those commercials as a kid of third world countries where children are starving, they have the big belly. it's not because they have a big stomach or big intestines, it's because they have a huge liver. Their liver's so busy trying to process proteins from their muscle or excess sugar that's in their blood and turn it into nutrients, that those fats are just swelling their liver. They're actually getting a very fatty liver. And then I mentioned this earlier, but if you accumulate fats inside of neurons, it slows down communication. So starvation can actually cause a fatty liver, which seems like it's not possible, but it does. Right. Another accumulation would be calcium, and I know you've seen this one before too. This is an example of calcium accumulating in a blood vessel. So here you can see the regular blood vessel and that dark accumulation of calcium. There it is up close. And here's the actual blood vessel. You know what this is? Here it loops around. It has a couple branches coming out this way and then a large section going down. This is actually the descending aorta and the aortic arch. But here you have calcified accumulations. The calcium gets really hard, almost like plastic. So if you were to squeeze on this area, you could crack it, just like a piece of plastic tube, a real thin plastic tube. Other accumulation, and they call this dystrophic calcification. Other accumulations may be in the lungs, like here you can see it building up between the alveoli, and sometimes just a hyaline change, what they refer to it, where it starts building up in other parts of the body. But this accumulation would be like an egg when you leave it on the counter for a day, or a little egg white on the counter. It turns like hard and almost crystallized. That's called glassy change or hyaline change. And then here's just another example of that hyaline change in the cornea. So this is accumulating calcium and it's starting to build up and it's clouding the cornea in this person. The last one, genetics. So genetics can have a huge impact on cells' ability to adapt or change. One problem that you can see with injury based on genetics would be sickle cell anemia. So with sickle cell, all it is one amino acid that's changed, and it causes the entire shape of the red blood cell to start changing. So here you can actually see a physical presentation of cell injury, and it's because of genetic structural changes. We'll talk about these when we get into genetics in just a few weeks, so I'm not going to go into it in much detail. And then the homework for this section is, right there. So homework question number 9, just go back through your notes, write down what the 4 forms of cell injury were, and just give me one memorable example of each. So it doesn't have to be detailed, just the 4 types, and give me one example of each.